Excellent. So I think we're ready to start. So hello, everybody. It's my pleasure to welcome you all to the Biomedicum Helsinki seminar. The seminar will last for about an hour. So 45 minutes will be the presentation, and then we will have about 10 minutes for the Q&A session. So during the Q&A session, I will ask you to use a microphone when you're asking the question, because the seminar will be live streamed, and everybody should be aware of what's being asked. After the seminar, there's going to be a student lunch for those who signed up, but we do have 2 free spots available, so if you want to join, get to me. And now that the practicalities are out of the way, I am happy to present Professor Yong-Fan Guan, who is an associate professor of the computational medicine and bioinformatics at the University of Michigan. Since 2013, she has written some of the best performing algorithms in the DREAM challenges, the largest system biology benchmark study. Professor Guan is awarded consistent Best Technical Performer Award for her group's achievements in the DREAM challenges and in the recognition of the open source software that they have contributed to the bioinformatics field. She is one of the few people internationally who owns multiple gold medals in the annual Data Science Bowl, and she has been published in leading journals such as Bioinformatics, GigaScience, Nature Methods Science, Nature Communications, and so on. So, without further ado, welcome.
Thank you. It's my great honor to be invited here to give this talk on machine learning in biomedicine. So my current research interest is primarily machine learning, while my previous training were in biology and mostly molecular biology. However, the growth of the field of artificial intelligence and machine learning have attracted many people's interest, including myself. And today, I'm going to talk about 3 projects that are either very closely aligned to the interest of the people here, or very recent that I think are very interesting technically to talk about. So the first one is drug synergy prediction. So this is just an overview of the problem. So often we have a bunch of chemical structures or drug structures, and we are given the pre-treatment genomic profiles, such as gene expression, copy number variation, mutations, and methylation. And we are asked to predict this drug synergy. So the reason that we are asked to infer the drug synergy rather than individual drug is that for cancers, the biology is very complicated, and often a single drug may not be sufficient to treat the cancer. And when the relapse come back again, it will be very difficult to treat. That is where the synergy combination of drugs come into play to address this problem. So the idea is that the multiple drugs or 2 drugs have to work better than what is expected from a single drug. For example, if we increase the dosage of a single drug, we could expect that we will see an increase in drug response. And by plotting those 2 together, we can, and according to some kind of combinatory estimation. So there are, in literature, there are different types of estimations of the combination effect. We can plot this overall curve of a expected drug combinatory effect. However, we could also actually put the 2 drugs together onto the cell line and observe at the intersection the values of this curve. And the difference between the 2 planes are what is being called drug synergy. So a better drug synergy would have a better drug effect compared to as if we applied them individually. So why is this problem important? So let us look at this matrix. Suppose we have 100 drugs and we have 100 cell lines. This will become a huge search space. 100 multiply 100 and multiply 100. So this will become 1 million combinations. And it will become something that is very difficult if we were trying to do it purely experimentally, a lot of work. And if we have sort of computational ways to prioritize what are the most possible points that we observe a good drug synergy response, it can save us a lot of money to find out the effective combinatory drug for cancer treatment. So in recognition of the importance of this problem, the DREAM Challenge, this is 2016, they organized this AstraZeneca-Sangor drug combination prediction challenge to benchmark the algorithms for drug combination prediction. So DREAM is a platform for more like data science. It is just one of the many different platforms, many different platforms for this. And it focuses on biomedicine. So in this challenge, we had 86 cancer cell lines, about a little bit over 100 drugs, and 11,000 drug combinations. About half of them, a part of them were used as training, and the other part were used as a blind test. So the idea of those type of challenges that you try to estimate the accuracy in a more fairly and blind way. So fairly in some sense that most of the participants in this challenge are 10 years younger than me. So it's not entirely, I'm just much older than them. That's why we performed acceptable in that challenge. And so by certain measurement that the accuracy we achieved in that challenge achieved the accuracy of the experimental replicates. That is what will happen if you do the experimental twice and calculate the correlation. It wouldn't be perfect. So of course this statement has a lot of constraints that we were tested on standard cell lines and very well studied drugs, which is an extremely easy problem to begin with. So on this problem, there's still a lot of space and a lot of open areas for us to explore. And actually, this accuracy isn't anyway from ideal, because an ideal accuracy would be the correlation between a pool of experimental replicates rather than cross replicates. So let us take a look at what we have in this problem and what is unique in this problem compared to many of the other machine learning problems. So to predict the either single drug response or drug synergy, we have a lot of input data and domain knowledge, and it runs through a certain type of machine learning. So what is machine learning? I guess this audience, most of you are very familiar with the concept, but I like to just interpret machine learning in the view that how I see this term. So this is a primary school math, that knowing X and Y, trying to find out A. This is what my daughter is doing right now. And if we go a little bit more complex, that we have a matrix of X and a vector of Y, and we want to find out the vector of A. Then this time it becomes a regression problem. So then what kind of a regression actually goes into this problem doesn't really matter, whether it's linear regression or lasso or ridge or support vector machine or Gaussian process regression. So they are all doing this kind of calculation that tries to find out the parameters, and all based on a variation of least square. So of course, how least square comes from into history is also coming from a competition where at that time people trying to find out the position of the stars, and Gauss correctly predicted the appearance of Ceres by the invention of least square regression. So competition is nothing new, challenges is nothing new in the history of science. And obviously by that competition, he successfully competed over several variations of least square, or the variations of currently used normal distribution. Such as the Laplace distribution were out during that kind of competition. And going a bit further, that if we know the matrices of X and the matrices of Y, and we can find out the matrices of A. So this becomes a multitask learning, right? So the overall machine learning framework, I think is basically what is being presented here. So what we can see that is common in the current machine learning framework that there's a step of dimension reduction. So it's trying to compress the information. And I think that is the essence of machine learning and also human intelligence, which is to compress the information, very complicated information that we accept in this world. For example, when I come here today,
could you maybe use this microphone?
It's not loud enough. Okay. Sure, certainly. Is it better? Okay. So, for example, I come here today and I see many people, observe many things, but often what we can summarize my experience is just one word: I'm happy or sad, right? So human intelligence is trying to compress information. So is machine learning, the process of machine learning. And then let's come back to see the problem of drug response prediction. We will see what is missing here. So we start with a matrix of cell line and genes. So this is the expression matrix, right? So this is a 2D matrix. And we end up with also a 2D matrix. That is the cell line versus drug matrix. Then there's something missing here because there's nothing to compress. We come from 2D and end up with 2D. So I think that's the most challenging part. The reason is that this 2D matrix is the pre-treatment profile. So this is there's a logic here that if we already have the observation of the post-treatment, then there's no point in prediction. So all we have is the pre-treatment profiles and try to infer what will happen after the drug treatment. So the idea that of our algorithm is to extend this 2D matrix into a 3D matrix through this gene-gene functional relationship network, and then compress this 3D matrix along the gene direction and leave us with a 2D cell line versus drug matrix. So this is a simulation step. It extends along the drug direction. What it does is it simulates the genomic profiles after drug perturbation. Because we do not have this observation, then our best guess is to simulate a third dimension of drug response. That is the expression after administer each of the drugs. And then we just compress against the gene direction. Exactly what algorithm, whether it's SVM or random forest, that we use to compress against the gene direction actually doesn't matter that much. What matters is that we use a space of simulated feature. I think this is a kind of a new idea. So now let me explain how I use this gene-gene network to simulate this space. So gene-gene network is the one that we used is a probabilistic network that indicates the likelihood of two genes working the same biological pathway. So the higher the probability is between the two genes, the more likely they work in the same pathway. It's a probabilistic connection. So our assumption is that for the direct target of the gene, here, their expression is turned to 0. Of course, this is a very coarse simulation, but it is better than nothing. So the target turns to 0, and the rest of the genes, the closer is the gene related to the direct target of the drug, the more its expression is being suppressed. So that's our overall simulation scheme. Of course, there's a lot of things to be improved here that whether this is exactly in correlation with whether this is in exact correlation with the network connections, or whether we should give a complete suppression of the target gene. So those are all open questions. This is really the beginning of the this kind of method and what we have so far. And through this simulation, it generates me a totally new profile of each cell line, which represent what would happen after the drug perturbation. And these simulated features were put into the base learners to generate a prediction for me. So the above were actually works that were done 2 years ago. I just feel a little bit embarrassed to still talk about quite old technology. So this is what is being going on in this field. My group personally is actually a bit slow in this area, but we can see that this is the right direction to go. So the chemicals are naturally a graph because of the connections between the atoms. So deep learning convolution network is learning spatial or time continuity. So the connections between the atoms naturally creates a graph. So nowadays a lot of people naturally creates a path of connections. So a lot of people are exploring this idea of using convolutional network to derive the features, hidden features from the chemicals. So the convolution is actually done on the connected atoms. So extending from that idea is that the gene-gene network is also a graph. So this could help us to naturally build the chemical graph together with the gene-gene interaction graph, and then use our simulated feature method to predict the drug combinations. Of course, this is all kind of future work that is that we will spend time to investigate. And as I said, we haven't really got anything that is really done yet. But let's suppose that we will be able to get it done. So this network will have some very beautiful properties that, for example, one thing we could use it is to find out the biomarkers within by this method. So this is a bird, and deep learning has this property that we could reversely going back to the network and look at the first layer gradient of the network, which is called the silencing map. The silencing map indicates the original features from the input that are most indicative to the output predictions. For example, I have a bird here, and what makes me predict that this is a bird? It is the eye and the beak. So this is where it is being highlighted. Similarly, if we could use this framework, it will help us to identify the atoms that are most important to this prediction, and the network modules that are most important to this predictions by the saliency map. So this is one of the future works that we'll be doing. So that is the project on the drug synergy prediction. The next project will be very, very different, but I think very interesting. So I'd like to share this with you. So this problem is about using the cell phones to detect Parkinson's disease in general population. So everybody uses cell phone, and one thing that not everybody knows is that in every of those cell phones, there's a built-in accelerometer that measures the changes in speed, and a gyroscope. So this is a gyroscope, this one, that measures the rotation rotation of the phone. So every cell phone, we could collect those data, the user acceleration, accelerator, gravity, and the rotation rate, which is the acceleration of the rotation, and attitude. So as you know, Parkinson's disease is represented by tremor, so they shake, right? And at a certain stage, Parkinson's disease, some of the Parkinson's disease patients will also develop freeze. So tremor is when you do not want to move, you cannot control your movement. And the freeze is more like when you want to move, you cannot move. So it's kind of an opposite process. And in the data that went into this project included resting and walking. So as you can imagine that in resting, what we want to detect in the Parkinson's patients is the tremor, because it's involuntary movement. And during walking, what we are trying to detect is the freeze. It's one sudden freeze, like walking in the middle and cannot continue to walk on. So now we can actually have a guess of whether resting or walking would perform better in terms of detecting Parkinson's. So because during walking is a combinatory movement of voluntary movement and involuntary shaking or freeze. So the voluntary movement will become a confounding factor in the predictions. So we could actually guess that for resting, it's very isolated. It's just predicting the tremor. So for resting, we can perform much better, and we'll see it's indeed the case. The other thing that we can, without doing this project, but we can try to guess is whether accelerometer or the gyroscope would perform better, or the rotation would perform better. So accelerometer is like when you consider the object as a point, as a mass point. So only the movement in the space could contribute to acceleration. But for rotation, even I hold the phone still, it can still rotate around itself. So it is much more sensitive. Even I do not move in space, it will have signals over there. So if you have some app on your phone, what you could see is that even you blow into the phone, you will see a signal in the gyroscope, while the accelerometer is almost stable. So we can actually infer that the gyroscope will work much better in terms of predicting the Parkinson's, and this is indeed what we observed in our experiments. Oh, so this is also a dream challenge that we did last year. So, so basically what we used is a deep learning framework that takes in the acceleration or the gyroscope information and use the walking or the rest data to predict the Parkinson's disease. So what is deep learning? Maybe many of you are very familiar here, but just let me briefly give a summary. So here we have this time series data of, this is like a millisecond, and we have the XYZ, which are the acceleration at each direction. And what deep learning does is to extract the locally connected information. So it's a local algorithm. That's why we always say that space or time continuity. So if it's discrete input, such as gene expression, individual genes, that lacks specific structure, it's very difficult to apply deep learning on it. But anything that with this continuity, this is time continuity, can be used in this deep learning through these convolution blocks that gradually extracts information from the continuous time frame and shrink the information to generate a prediction. So again, this is a compression process. So everything in machine learning is just a different way of compression. This is shrinking the original time space. So everybody nowadays knows deep learning in the machine learning field, that what makes our algorithm that stand out is different kind of strategies. Obviously, the normalization. So if I hold this phone horizontally and versus vertically, the XYZ values will be completely different. So one of them, the X, would be at 9.8, that's the gravity. And the other way would be the Y, would be the gravity value. So normalization across each of the axes is essential. And the other way is pooling. So we put the maximum prediction value across multiple tests for each patient. The reason is that even for patients, they do not necessarily shake all the time. It's their most severe time that determines this person is a patient. And the most interesting part, I think, is the augmentation that we did on this data set. So data augmentation is a technique that used to combat overfitting in deep learning. So deep learning is very, very powerful, but it has a strong limitation that it remembers everything it sees. So what will be remembered in this network is actually the location of the person that takes in the record. So what the deep learning will learn is whether this record is taken in California or Europe. That's a very sad thing. So what we need to do is to recreate this record as if it is taken at different positions on the Earth. So this is letting the network to see many, many different types of data. So before I go into explain how we did the augmentation, so let us first see the effect of the augmentation. So this is about overfitting. Without augmentation, this is number of epochs. This is the training error, and this is the testing error. So the training error quickly drops, but the testing error increases just almost immediately after my training. This is because the system starts to remember all the examples that it has seen. And then I will perform very poorly in a new example. But with augmentation, we could see that the training error and the testing error drops together with the training error. And I think what is most important in machine learning, in my perspective, is probably just to adapt a very standardized algorithm into the biological insight, or in this case, the physical insight of the problem. So this is one physical aspect of the problem. So the cell phone, the reference frame can be different for each cell phone. So for every of your cell phone, the first time when you open the cell phone, it determines a reference frame, which points to this side as X, and this is Y, and this is C. This is Z. So then whatever the reference frame is would completely affect the number, the numerical value recorded in the accelerometer or the gyroscope. And also when I hold this phone here versus I hold this phone in this direction or at any locations of the Earth will create completely different types of record, numerical values, which is irrelevant to the Parkinson's disease, which is irrelevant to the disease or not. So the way that we try to combat this is to start to create new examples from the original example by rotating the reference frame of this phone. So the way that we did this rotation is actually already being generated by mathematicians, Euler, 200, no more than that, probably 400 years ago, hundreds of years ago. So his method is that for any object, the rotation, when I want to rotate the reference frame, the rotation can be expressed by a single vector. So this is the axis. This is this axis. This axis. And the rotation can be expressed by this axis and the angle across it. So this is the angle. Oh, whoops. So any rotation can be expressed by this. What does it mean? So like the Earth rotation can be expressed by an axis from the center of the Earth to North Pole, right? And we are just rotating across that line, center to the North Pole, and there's always an angle across it. That's the Earth's rotation. So, and any kind of rotation could be expressed that way. So by this formula, we could create this rotation matrix. So this rotation matrix generates new reference frame, which can be directly multiplied to the original record. This multiplication generates me a new record as if the record were taken at a different place on the Earth. So what we do is we did a 360-degree augmentation of this record. So although most of the data of this study were collected in certain places of the Earth, mostly in US, it helps me to generate examples across the Earth. The other augmentation that we did according to the nature of this problem is this time-wise scaling. So we do this because some people could walk faster or slower. It doesn't, or they shake faster or slower. It doesn't really mean that there is a significant difference between the pathology. Of course, this scaling cannot be extreme, right? So there's it has to be within certain range. So what we did is 0.8 to 1.2 time-wise scaling. It's like adding a wave to the time axis as a perturbation of the original data. And also the scaling on the magnitude-wise. So some people have a bigger terms of the shaking, and others are smaller. And this again is something that's related to the pathology of the disease, and we have a limitation of how much we actually do the scaling. So let us see the differences in the performance in terms of augmentation versus not augmentation. I think most of the other teams were using very good deep learning networks. Actually, personally, I don't know how to tune those networks at all. So I think the main difference comes from this augmentation, that it increased, it boosted our performance quite significantly. So as I mentioned earlier, so on the walking, the AUC was 0.87, and on rest it is 0.9 because of the confounding factors in the walking data. Okay, so the previous project was about an end-to-end prediction of a disease. So in the recent years, there have been a technique that doesn't just do an end-to-end prediction, but give you a prediction of, for example, every time point in your input data. So I think this is a really growing technique that has been already very well studied in the image field, but not sufficiently applied to the biomedical field. So that's why I included a third project, which I think will be quite interesting to even genomics. So you can do like nucleotide-wise segmentation, right? So this is an idea that I'd like to talk about. So this problem is about detecting the sleep arousals. So everybody, not everybody wake up in sleep. And the pattern of healthy individuals and diseased individuals of arousal are often very different. So the people with certain disease problems can wake up more often and more irregular. So there are several different types of sleep stages. And the spontaneous sleep arousals is defined as the transient intrusions of wakefulness into sleep. So it's a very natural kind of brain activity change. But if you have too much of those sleep arousals, obviously you will have daytime sleepiness and also sleep disorders. That's why we're interested in this problem. So as you can see, this the yellow part in those figures, those are different individuals. The yellow parts, the yellow spikes here are the arousals. So this is the part that we are interested to detect. So this is the input data. So it has the brain signals, the ECG, the EEG, and also the movement record. So the movement of the abdominal, chest, and chin. So all these records are taken at 200 frames per second. And the question is, how can we predict the places where there's a sleep arousal based on a parallel of all these records? And this is a technique that we will use, which is called translational network. So let me just explain a little bit what is translational network. It can do a lot of different things. So this is me when I was little. And at that time, we didn't have colored images. We didn't have colored photos. So this is what I do today, that I color it myself. So this is a translation network that we trained to translate black and white. The input is black and white into output of a colored image. This is a translation, right? So the size is the same, that is translation, but it's just the content is different. And this is another translation network. This is what I paint. Of course, it's very mediocre, but let's change it to something more prettier. So this is a translation network trained on Van Gogh's paintings for 300 iterations, and it starts to appear those curvy, very specific type of strokes that were done by Van Gogh. And this one is the one that I like the most. This is Monet's, trained by Monet's collections, and it gives a more dreamy, impressionist appearance. Oh, this one is trained by the Dalton's watercolor collection. So it actually gives a super resolution. So translation can give you a resolution that is higher than the original. And in this painting, what is being reflected more is the watery, the light and the watery, very characteristic of his work. So this is also translation. It translates a style. This is also translation. So this is the project that I did for one of the data science balls. So what is being translated here is a isolation, is a segmentation of the image. So the input is this X-ray of the hard part is this way, and the output is a segmented left ventricle of the heart. So this is also translation. It translates the input image into a binary mask. So this is also translation. So this is trying to detect breast cancer. So these ones are tumors, and these ones are calcification dots. So it can give you so what is being presented here is that it tries to give you a pixel-wise isolation of the interested region. So we can give a pixel-wise isolation of the interested region here, then we can give a millisecond-wise isolation of the sleep arousal later. It's the same idea, except that this is in 2D and that one is in 1D. So this is last year's data science ball gold medal solution. So this one is to segment and count the number of cells. It can also give the classification of the type of the nuclear. So again, this is another translation. It translates the input image into another image of the exact same size that gives the individual localization and segmentation of the cells, cell nucleus. So now let's take a look at our overall scheme of the solution. So the UNet is just one variation of the translational network, but I feel this is just a very elegant way of representation of this type of translation network. So it consists of two parts, the encoder and the decoder. The encoder part encodes the information that takes in, whether it's an image or multiple signal input of the brain or whatever. So the input size will be the same as whatever will be decoded. So the encoder extracts the hidden information from the images or from the signals, and shrink it in the size-wise direction, but extend it on the feature-wise direction. So the overall information is preserved. And then the decoder translates to whatever you want to be outside, whether it's a segmentation of the arousal, or whether it's a segmentation of the image, or changes in color, or transferring styles. They are all the same. Essentially, in terms of machine learning. So this is the translation process. First shrink, and then recover to the original size to give you the prediction of the arousal state, arousals in your sleep. So this is the final performance that the AOC we achieved was 0.92, and the AOPRC was 0.55 because the arousal event was very, very sparse in the original data set. So that is actually my last slide. So any questions?
Thank you very much. That was really interesting. I have a question, actually.
Go ahead. It doesn't.
Hello. No, that works. So I have a question. You seem to be doing a lot of deep learning. Could you maybe comment a little bit on what you're using for the computational part of things? What sort of resources? Is it cloud computing? Is it on-site computing?
Mm-hmm. So I'm actually a video game person. So before, initially we just used, actually until this point we were using game cards. And all the ones that finished the deep learning were going to basement for gaming. So the computation that were so far used in computations were pretty limited, actually. So I usually assemble most of the servers myself with just a regular desktop type built with two NVIDIA cards. Since this has become from a hobby, this was really hobby, into a profession, and we just recently built an 8 GPU cluster. That is a more professional, it's still gaming card actually, but it's a more professional way of dealing with the deep learning. So other than the GPUs, one thing that is pretty critical, I guess, is the SSD access for the reading to the, because the reading from the disk will be a limitation. So that's the main two parts. But it's really just very regular machines. So far, we're using machines within $4,000 budget. Yeah, but we're going to have some good ones since we're going to do some serious research in this on this aspect.
Cool, excellent.
Mm-hmm.
No, it's better if you're using a microphone so that also the people who are watching the stream can hear the questions.
Thank you very much for the interesting talk. I'm not from your field at all. I have a tricky question for you. Have you ever tried to give a Parkinson's patient a mobile phone into their hands? These Parkinson's patients very often don't even can use a spoon to eat. Mm-hmm. So are you fixing the phone on their hands with some tape, or how do you measure these things? Parkinson's patients
I see. That's a very
really
good,
have
that is
tremendous problems to use a phone. They usually don't use them.
Mm-hmm. That is a very good question. So, so this algorithm is more for population-wide screening. It's for early stage of Parkinson's. But there is indeed further development because when our algorithm is delivered to the patients, care, and also to the pharmaceutical companies, they are more interested in severe patients, right? So there's another way that we do this, although they cannot carry the phone to walk around, or so there's a voice thing that you can just say, and detect this is also another deep learning algorithm that we could use the to detect the Parkinson's severity. That's more like a severity prediction. And there's another one for even more later stage patients, that is for tapping. So it asks you to tap on the cell phone. And the way of tracking the tapping together with the voice is a better way to track the severity of the disease. Mm-hmm. Does that answer your question?
Partially, yes.
Mm-hmm.
Parkinson's patients can relax, but if they need to do a task, they start to shiver, they start to shake. So if you tell them that now tap on the phone, then they get the problem. Yeah,
it is really true for the really kind of bad bounded patients. Then above certain level, like on the UPDRS score between 0 to 5, above certain level, it will be very difficult to do any testing on them. Mm-hmm. Thank you for your question.
Sorry, there is one more question, I believe.
Two even maybe. So when you were trying to predict drug combinations, you mentioned that you use gene expression profiles of the cell lines that are just growing, I guess, no drug treatment or anything. And then you have the data how the single drugs affect the cell line. And then you're doing modeling how gene expression would change when the drug would be added. But what kind of data do you use to model this? What are the assumptions to make that modeling? You need to have some kind of start or some assumptions.
So I guess I didn't explain that part very clearly. So the assumption is just that the drug target will be affected. So we need to know the drug target. This is really just for known drugs. There are a lot of things to be done in this field. The drug target will be affected a lot. That is one assumption. And the genes that are more closely related to the drug target will be affected more. So the drug target expression will be turned to 0 no matter what. So we have the baseline profile, right? So we know like this gene expressed at probably with a value of 5, we turn this 5 into 0. And for another gene that we know that is closely related to this direct target, has a value of 4, and the gene is related to like 0.5 probability, then we turn its value to 2, because it's 2 multiplied 0.5. That's the assumption.
So you're
behind
using
it.
a lot of correlations from published studies.
Yeah.
How did, okay, yeah.
Yes,
that
the
was-
correlations between published studies, essentially, yes.
All right, and second question is related to that Parkinson's, because you said this is more for population-wide. I'm sure insurance companies would be very interested to see whether the patients or the people that are trying to get an insurance from them will have a chance of getting Parkinson's. And unlike, for example, sequencing your whole genome, you need to consent to that. But here, that algorithm could be put into any app, I guess,
Yes.
and access to the gyroscope and that other thing, and the insurance company would know if your prediction is really good, whether the patient or the person will have Parkinson or get Parkinson. Is that a concern at all?
Yes, so right now FDA on the digital health part hasn't, I don't know what's the regulation in Europe, but in US it's usually regulated by FDA, right? So they are still in the stage of developing protocols for allowing those apps to go into even for just the general use by people. Monitoring is usually fine, but connecting to insurance company, of course, this is personal health data, and there's indeed some concern on it. Because this is really a new area, the policies is still being developed, and they just changed the commissioner, so it's difficult to see what will be the next step, what will be the allowance yet. Mm-hmm.
I guess that will be the last question.
Hi, thank you for the nice talk. So I have a general question so with regard to machine learning. So you were showing how machine learning can be used to predict so you use some information to predict, like for example, synergy, response of different drugs and things like that, sleep arousal. So it's mainly being used for this prediction purpose. So do you think, or what are your thoughts on whether machine learning can also be used for scientific purpose to understand underlying mechanisms, underlying different diseases, and so in terms of getting some understanding of the underlying mechanisms, rather than just to predict whether a disease is happening or not, or this kind of thing?
Yes, so machine learning, people always say it's a black box, right? So yeah, I used to introduce machine learning like a box, and I will say this is done on purpose, a black box. So the thing is that we do need biological expertise to help us to understand algorithms. So for example, for drug synergy prediction, there's a whole branch of different machine learning just for finding out the biomarkers. It's not the ones that I have been talking about. But even we find out that we need a biologist help to help us understand. Of course, there are a lot of issues with multiple testing, etc. So right now, I think there's a disconnection between the experimental side and the computational side. They try to collaborate, but there's always this mentality that on computational side, we just saw that if my algorithm works great, then it is great. If an algorithm that with great biological insight but just doesn't perform well, what's the usage of that algorithm, right? And for biologists, they always thought that, okay, this has to make sense in certain biological pathways, but bear in mind that those pathways were biasly studied in literature. No matter what, at any stage in human history, our science is completely biased, right? So, so there's always kind of discrepancy between the two. I think one thing in common is that they try to justify what they are doing is correct. That is the common mentality. So in the future, when the benchmark people and computational people work more closely to each other, I'm pretty sure that there will be a spark in terms of sudden spike in discovering new biology using the AI technologies. This is something that we will see in the next 10, 20 years.
Thank you.
Excellent. Thank you very much, Professor Guan. Thank you very much, the audience. Those who want to take part in the student lunch, please get to me. And yes, thanks a lot.
Thanks. Nice talking.